Hello, everyone. My name is Juan, and myself, Tina, and Lucero will be talking to you today about Klebsiella pneumoniae.
Why do we care about these superbugs? Superbugs are a strain of bacteria, viruses, parasites, and fungi that are resistant to most of the antibiotics. Klebsiella pneumoniae carbapenemase, or KPC, is highly drug resistant, gram-negative. So the carbapenem resistant are a family of bacteria difficult to treat because they're highly resistant to antibiotics. Carbapenemase is an enzyme produced by the bacteria, and this breaks down the carbapenem antibiotic. So in this infection, the infection control is very critical. Okay, KP can lead to dangerous clinical manifestations, like, for example, organ failure, pneumonia, sepsis, wound and surgical site infections, liver, respiratory tract, bloodstream, and other infections. Each part of the body is different, so each is going to have its own symptoms, and this bacteria is has highly mortality rates and morbidity rates. All right, Klebsiella pneumoniae is a gram-negative again, and it's normally inhabited in organs and feces. And the infection can be viewed as a consequence of specific interaction between pathogens and the host. So this involves early interactions with the innate system. And the models usually assess the Klebsiella infection has been done by infecting rodents, specifically mice. So neutrophils are critical component of the innate immune response against bacteria because neutrophils remove bacterial pathogens through a process known as a phagocytosis. And also the neutrophils express several groups of receptors that can induce the formation and generation of reactive oxygen species known as ROS. Um, Also in the adaptive immune system, we have the beta microglobulin with B2M, which is a component of MHC class 1. And also the B2M is critical for the immune adaptive system because the dysregulation of B2M is associated with multiple diseases. Specifically, CD2 T cells play an important role on the adaptive immune response.
The innate immune response to bacterial organisms includes phagocytosis by neutrophils. So here in figure 1A, these black arrows are pointing at KPC, and this is the neutrophil up here. And this is indicating that KPC evaded phagocytosis, whereas in 1B, the arrows are pointing at Staphylococcus aureus, and you can see that it's inside of the neutrophil, indicating that phagocytosis was successful. And a phagocytosis assay can be used to determine the total number of bacteria ingested per neutrophil over a period of time. So in figure number 2, there was a Staphylococcus aureus bacteria used as a control because it's known to be readily ingested by the neutrophils. And there were also 6 KPC strains that were used, and KPC again are the ones that are exhibiting antimicrobial or antibiotic resistance. So here you can see that the S. aureus strain was readily phagocytosed over time, and that's indicated by this line that is going up. However, all of the KPC strains are way down here, meaning that they showed resistance to phagocytosis. Neutrophils are also classified as granulocytes, which means that they are capable of degranulating. And one of the components that they release are reactive oxygen species. And degranulation will occur after phagocytosis. So here in figure 3 is a graph that's showing ROS production, ROS meaning reactive oxygen species, over time. So there were again 6 KPC strains that were used and compared with a zymosan control. And zymosan is just a component of a yeast cell wall. So we can see here that ROS production for the zymosan was increasing over the first 30-minute interval. However, the 6 KPC strains showed negligible ROS production. These results are actually consistent with the previous slide showing the phagocytic index, because if there's no phagocytosis that's happening due to their resistance to it, then we wouldn't expect to find elevated levels of reactive oxygen species. Antigens originating from pathogens or intracellular pathogens are loaded on an MHC class 1 receptor. And this can be recognized by a CD8 plus T cell for activation once it recognizes and binds the antigen and the MHC.
Okay, so as Juan mentioned before, we do have an MHC1 receptor. In this receptor, we can find a beta-2 microglobulin or beta-2M component as shown in the image. The CD8 positive T cells do create an inflammatory response upon that antigen recognition. These CD8 T cells need to bind to the MHC1 receptor from an antigen presenting cell. Okay, so here in figure 4, we have 2 graphs where we are looking at the colony forming units in the spleen and liver for both beta-2M knockout and wild type mice. In the figure, the black bars represent the beta-2M knockout mice and the white bars represent the wild type. We are looking at the colony forming units in the 12th and 24th hour mark. And we can see that in both graphs, the beta-2M knockout mice have a higher microbial load for both, in both the spleen and the liver. Seeing this, we know that mice do not survive in the absence of beta-2M. Okay, so to recap, KPC is resistant to phagocytosis by neutrophils, and beta-2M deficiencies do impact the adaptive immune response because it's part of the MHC1 receptor. This MHC1 receptor is required for CD8 T cell activation. The research does indicate that if beta-2M component is not present in the MHC1 receptor, it does increase the susceptibility to bacterial infections, letting us know that a non-functioning MHC will end up having more viruses. KPC must be presented to CD8 T cells for effective clearance. KPC will exhibit a higher mortality and morbidity rate among susceptible patients through bacterial infections. These KPC infections do have limited therapeutic options due to the fact that they are highly resistant to carbapenem antibiotics. Carbapenems are known to be quite powerful antibiotics. Since KPC is highly resistant, it is able to transfer other bacteria easily. Some individuals have to even resort to archaic antibiotics, such as colostrin, which is known to be a very toxic antibiotic.